Signal Transduction Presentation by Jane Esala. Cell cycle regulation in the cell. Cell division happens from the time an embryo gets its start through adulthood in eukaryotes. Cell division also happens in all cell tissues at one time or another. Once a eukaryotic organism has reached adulthood, typically, cell division is dormant unless it is triggered by an outside source, or there's a defect in the regulatory mechanisms that limit cell division. When these mechanisms are not regulated, cell division will become uncontrolled and can lead to cancer or other disorders. The biochemical events needed to create new cells must happen in a precise order. Signaling mechanisms are required to regulate cell division. Before we move into the regulation of the cell cycle, let's do a quick review of the steps of the cell cycle. So the cell cycle is a series of specific events that must happen in a specific order. The process will produce two new daughter cells that are identical to the parent cell. The steps are specifically timed and monitored. And we have two major phases. We have interphase and we have the mitotic phase. And in interphase, cell growth and DNA replication happen. During the mitotic phase, DNA and the contents within the cell will separate and the cell will divide. Here is the entire cell cycle from start to finish. In the gray, we have interphase, and it's the largest portion of the process. Within interphase, the first round of cell growth and the DNA synthesis portion of the process take up the majority of the interphase time period. Mitosis and cytokinesis wrap up the process with the production of the two new daughter cells. In interphase, the cell will continue to go about normal cell processes. The interphase steps must be completed in order for the cell to start the mitotic process. In the G1 phase, which is also known as the first gap phase, the cell starts to gather the building blocks of chromosomal DNA and proteins. At this time, the cell will also gather energy reserves in preparation for future phases. The S phase, or the synthesis phase, is the time where two identical copies of chromosomes are made. At this point, spindle fibers are also beginning to form from the centrosomes. During the second gap phase, or G2 phase, proteins needed to proceed through the cell cycle are synthesized. Organelles are duplicated and the cytoskeleton starts to break down some so that it is prepared for spindles to attach. The M phase, or the mitotic phase, is the process of the cell cycle in which the cytoplasm and genetic material are divided. In prophase, which is the first image here on the screen in this line of the mitotic phases, chromosomes will begin to become visible and the spindle fibers begin to appear. You can see the spindle fibers attached to the red dot here, which is the centriole, and those spindle fibers are preparing to attach to the sister chromatid in a later phase. In this prophase, the nuclear envelope also begins to disappear to make way for our chromosomes. The centrosomes move to opposite poles of the cell. In the next phase, our prometaphase, chromosomes condense further and the kinetochores are found on the centromeres. During metaphase, our third step here, you'll start to see the chromosomes line up in the middle, which In this image, almost appears like red X's, but those are our chromosomes. And you can also see the spindle fibers stretching out from the opposite poles and attaching themselves to the sister chromatids. So the sister chromatids will become attached to the spindle fibers, which are at the opposite ends of the cell. In anaphase, which is our fourth phase here, the spindle fibers are pulling those sister chromatids apart and moving them toward opposite poles of the cell. And the cell will also become more elongated at this point. During telophase, the chromosomes have moved closer to opposite ends of the poles, and now we have a nuclear envelope surrounding both sets on opposite ends. Spindle fibers will start to break down, but they will still be pulling those opposite poles further away from each other. After these processes have ended, we start cytokinesis, and cytokinesis will pull the cell apart into the two new daughter cells. External events can trigger initiation and inhibition of cell division, and this can happen at the time the cell is ready to start replication process. Events that can trigger inhibition and initiation can be simple. So, a couple of examples may be nearby cell death or a growth promoting hormone that has been released and can be a trigger. HGH, which is the human growth hormone, can inhibit cell division if it's lacking and can cause dwarfism. Too much of that in HGH can cause gigantism, and if cells are too crowded, this can also inhibit cell division. No matter the reason for the signals, cells receive these signals, and this begins a sequence of events that allow the cell to continue into interphase. At this point, in order to move on, every factor required during each phase of the cell must be met. As most of us have learned in basic biology, daughter cells are typically exact duplicates of the parent. However, mistakes can and do happen when copying or dispersing chromosomes. This causes mutations that get passed on to each new cell that is produced from the abnormal cell. In order to prevent the abnormal cell from continuing to divide, internal mechanisms are working at checkpoints throughout the cell. Within the cell, the checkpoints exist at different points that allow the cycle to stop until conditions are favorable. These points happen close to the end of the G1 phase, at G2 and mitosis transition, and in metaphase. Whether external or internal activities, the cues that are given change the core cell cycle regulators within the cell. These regulators can be the reason for key events. For example, DNA replication and the separation of chromosomes. In the following slides, I will discuss these regulators and how they impact each phase of the cell cycle. Cyclins are some of the most important core cell cycle regulators. There are 4 basic cyclins in most eukaryotes. These are G1 cyclin, G1S cyclins, S cyclins, and M cyclins. Each of these are related to the specific phase or transition that they help to propel in the events of that phase. Each level of the different cyclins will vary throughout the cell, and as seen here, each cyclin is low at majority of the cell cycle, and then the levels will increase when they're needed. For the cell cycle to continue to move on, target proteins need to be activated or inactivated by cyclin. Events in the cycle are driven by cyclin by working with enzymes known as cyclin-dependent kinases, or CDKs. A cyclin by itself is not active, but when it's bound, it becomes activated and becomes a functional enzyme. This means that it's able to change target proteins. Specific target proteins are phosphorylated by CDKs. The phosphate group that is attached will make a target protein less active or more active. Once the CDK has cyclin attached, it will become activated as a kinase. Cyclin will also guide the CDK to specific target proteins that are the right ones for that period of the cell cycle. For example, CDKs will be sent to S phase targeted by G1S cyclins, which start DNA replication. CDKs will be sent to the M phase targets by M cyclin, which will cause the breakdown of the nuclear membrane. In the image on the left, you can see that there are no cyclin, so the CDK is not active, and no phosphate groups are attached to any of the targets. As a result, the S phase of the cell cycle is turned off. In the box on the right, the G1S cyclin is attached to the CDK. So the G1S targets have been phosphorylated, and this triggers the start of the S phase. So as a result, DNA replication enzymes are also activated. Cyclins and CDKs will work together to control the process of the cell cycle. The two together create a maturation promoting factor, or MPF. M cyclin levels will remain low throughout most of the cell cycle. When the cycle is close to G2 M transition, M cyclin levels will raise. CDKs that are present will bind with the accumulating M cyclin to form a complex. This complex will get ready to trigger the M phase, which will become active after the complexes get an additional signal. Phosphate tags are added by the complexes to proteins within the nuclear envelope. The addition of these tags will also cause the breakdown of the nuclear envelope, which is very important to the early M phase. Chromosome condensation is also encouraged. Here, the nuclear envelope breakdown by the MPF is shown. In the box on the right, you can see the M cyclin attached to the CDK, which will activate the kinase. The M phase targets become phosphorylated and the M phase will start. On the right, you can see the example here as it shows the breakdown of the nuclear membrane to allow access to the genetic material within. MPF also helps to push the M phase of the cell cycle. It also plays a part in its own demise by triggering the anaphase promoting complex or cyclosome or APC. This is an enzyme and protein complex that will drive the cell cycle out of mitosis by destroying M cyclins. This will let the daughter cell begin the G1 phase. APC also helps to break the proteins connecting sister chromatids. The separation of the chromatids let them move to the poles on opposite sides of the cell in anaphase. Ubiquitin is a protein tag that is attached instead of the phosphate group. This sends the target to the proteasome and it destroys it. Securin is typically bound to separase. When APC tags securin with ubiquitin, the securin is no longer able to keep separasin inactive. So separasin breaks up the cohesin, holding the sister chromatids together. So let's look at this in this image here. So in the purple at the top left corner, we have the APC, and the APC adds the ubiquitin to the securin, which is currently bound to separase. The separase is still inactive here, and you can see the cohesin holding the chromatids together. Now that the ubiquitin has been added to the securin will be sent to the proteasome, and the proteasome will act like a recycle bin and it will break it down. Now the separase is free to become active and it will break down the cohesin, and this will start the separation of chromatids. The regulatory processes discussed so far are what we would call positive regulation of the cell cycle. The positive regulators activate and drive the cell cycle to start, continue, and finish. Negative regulation will stop the cell cycle. This is done with regulatory proteins called retinoblastoma proteins, or RBs. These proteins are tumor suppressors. RB, P53, and P21 are three regulatory proteins that work to suppress the cell cycle. They regulate the G1 checkpoint, and if there is damaged DNA, P53 will stop the cell cycle and activate enzymes meant to fix the damaged DNA. If DNA is not able to be fixed, P53 will activate apoptosis, or cell death, also known as necrosis. This is done to stop the cell from creating new damaged cells. The levels of P53 will rise and cause the production of P21. The stopping of the cell cycle will be helped by the addition of the P21. In response to more stress, P53 and P21 levels will increase and make it more difficult for the S phase to start. So what happens when a cell regulatory checkpoint doesn't work? When the regulatory processes no longer function, cell growth becomes uncontrollable. This leads to mass cell growth, also known as cancer. The process that regulates the part of the cell cycle in which DNA is replicated is extremely important. This is because if there is a mistake made in the replication process in the S phase, there will be a mutation in the DNA. If that mutation affects the part of the DNA that codes for the proteins used in cell reproduction, the change could mean improper binding of CDK to cyclin or targets detaching from proteins. Each of these possibilities can lead to uncontrolled checkpoints throughout the cycle. Even a small change in the nucleotide sequence can lead to a larger problem. Small errors can be sped up or made larger in the next generation, and so on. As the speed of the abnormal cell growth exceed the cell growth of normal cell tumors will start to form. Proto-oncogenes are the gene that codes for positive cell cycle regulation. When they mutate, they cause a cell to become cancerous. These are called oncogenes. Oncogenes specifically lead to increases in cell cycle progression. So as we've seen here, cell cycle regulation is very important, not only to just making sure that each process within the cell cycle happens in order and when it's supposed to, but it also prevents the cell from having just unregulated, constant cell growth, also known as cancer.